Okay, so we can move to the next presentation, and I'm happy to introduce Guido Walter Di Donato that is presenting GenoGra.
All right, so hello everyone. I am Guido Walter Di Donato. I am a postdoctoral research here at NextLab, and I am the CEO of GenoGra, the startup that is born at the NextLab and that is building the first pangenomic analysis pipeline. So today we are here because we all know the importance of genome analysis and the potential impact of this technology in different areas of science, especially in that of personalized medicine, where genome analysis enable early diagnosis and targeted therapy in different medical fields like oncology, cardiology, and rare diseases. The problem is that despite these very good and promising results, today, only almost 7% of the patients can actually benefit of genome analysis. And this is mainly due to the fact that genome analysis does not scale very well. From our customer interviews, it emerged that it can take from 10 days to up to 18 months for geneticists to get back the results of genome analysis. And this, of course, is not compatible with the medical practice, but also other application of genomics. And if we go deeper in this genomic workflow, we can easily understand how the secondary analysis, that is the software-based analysis, so the one that is made through the computers, basically, is the main bottleneck of the whole process. This is due to the fact that the available software tools for genome analysis are redundant and error-prone and are very hard to use without offering any ease of usage to the users. This also comes with insufficient scalability of the analysis, also due to the lack of qualified personnel that can operate this tool that are too hard to use. So for all these reasons, today, we are not leveraging the full potential of genomics yet. And to understand why we are not able to do that today, we need to focus on the paradigm of the analysis that is implemented in currently available software. So current solutions are limited in scalability because they all involve comparing the DNA data of our sample with a reference genome that is the same for the whole the species, for example, for the human species. And this comes with a lot of limitations, both in terms of accuracy of the analysis and also, of course, of scalability, given that we all share more than 99% of our DNA. So we need to change the paradigm of the analysis. And starting from 2016-17, the research community emerged with a proposition. So to move from using reference sequences to reference genome graphs, so entering the era of pangenomics. So instead of using one reference sequence, we can use some pangenomes as reference, so some collections of genomes that contains genomes from various individuals of different kinds, with different features, so from different populations, for example. And this is able to reduce the bias of the analysis and also to reduce the redundancy of the analysis, because we can focus more on the variations that are the actually informative part. And in particular, we are able with genome graphs to represent genomic variability in the most, in the closest way to the actual biological reality. So we are able to represent very, in a very precise way, some structural variations, some copy number variations, and so on, that are very difficult to target with string-based softwares. So just to wrap up, the advantage of this new data representation is the great scalability, the optimal quality of the analysis, and the unexplored application that this kind of representation can enable. For example, studying how genomes evolve during time. So this is the reason why in GenoGra we are currently developing the first pangenomic analysis platform. It's a software platform that is usable both for research and clinical purposes, and that is completely based on genome graphs. So it implements this new paradigm of the analysis, and thus it offers some much faster and easier workflow of analysis that involves much less tools, while also being more accurate. And this is all possible thanks to our core technology. Our core technology is basically made of two things. The first part is a way to represent and store the genomic information together with all the data, the metadata that are associated to the sample, included in our reference graph. And the second part, that is the actual core of our technology, is a sort of search engine, a sequence to graph aligner that is able to map DNA sequences of interest onto the genome graphs in a very efficient way, leveraging hardware accelerators. In particular, we are using GPUs to speed up the process in a completely transparent way. And this is patent pending, both in Italy and at PCT level. So this technology allows us to be very competitive with respect to our competitors in the market on a lot of different factors that emerged as value drivers from our customer interviews. So, for example, the possibility to do population genomics in an efficient way, or having both high performance and the highest quality of analysis provided by among all the competitors. So this places us in a very competitive position in a market that is growing really fastly. The genome analysis market size, this relates just to software and services for genome analysis, is growing really fast. Is actually one of the fastest growing market overall globally. And it is, and forecasts are that this market will reach in 2028 almost 12 billion of size worldwide. We are focusing on the European market that is almost 25% of the total market, and today is almost 1 billion, and it is expected to grow up to almost 3 billion in 2028. But the most important thing is not how the market is growing. Of course, this is a positive thing, but the actual informative part is that the market is segmented in three main customers. So we have diagnostic applications, medical applications. We have base research. So we target, in that case, customers like university or public research centers. And then we have applied research, especially in the field of biotech and pharma. And our technology is actually so flexible that we can target all these markets, all these segments, but we also developed a go-to-market strategy to validate as soon as possible the technology on the market while reaching the most regulated market. So our plan is to start with the non-human research. So we don't need any compliance, GDPR compliance, and so on. We just need to protect the data of our users. And this is quite simple. There is this off-the-shelf solution to do that. So we just need to integrate that. Then, after getting the GDPR compliance and guaranteeing a safe processing during the whole stage of the processing of the genomic data, then we can move to human research. And finally, after getting the in vitro medical device regulation, we can get to the diagnostic market. Of course, to reach these diverse segments of customers, we also need a flexible business model that can adapt both to the user's need, but also to the customer's need, because in practice, these two figures are not the same. So as a user need, we need to understand which are the tools, the services, and the kind of interface and experience that is best suited for our kind of users. On the other hand, we need to satisfy the customer's needs. So the number of users that need to access the platform, but also the kind of deployment. So you want the deployment on cloud or on premise. We support both, but of course, it changes the subscription fee and our business model. So moreover, an important thing is that GenoGra is using an open core model. So basically, we not only develop some proprietary tools and algorithms that we integrate in our premium platform, but we also contribute to existing open source projects that then we integrate in our platform. We also develop utilities that we make available to the community. This is to provide also in a free way, in an open way, some contribution to the fields. And so to conclude, let me present the team behind GenoGra. I'm not alone in this adventure, and I'm lucky to have a great team with Alberto, Guglielmo, Mirko, the Professor Sant'Ambrogio, of course, and also Alessandro in the part of business development. And we also have two advisors that are helping us on a strategic point of view and on medical applications of our technology. We are a very technical team. Our competencies are very tightly related to the high-performance computing, the genome analysis, and the graph analysis world, and we developed our expertise working not only at Politecnico di Milano and at Next Lab, but also in collaboration with other institutions, university, and also companies, both in Europe and in the United States. To conclude the presentation, let me show the current status of GenoGra. We are right now, after 3.5 years of research and development, we are at TRL 5. We have an MVP that is available. So the platform, the prototype of the platform is available and has been already validated by 2 geneticists. Right now, we are looking for seed funding to reach TRL 8. So we are looking for 600K. But of course, I'm not here to look for investors. Of course, if you want to invest, just drop me an email. But the reason why I'm here is that to reach TRL 8, we need to validate our technology. So if you process genomic data, if you are working on some application involving genomics and genomic data analysis, then maybe we can be a partner to do some pilot study where we can use our technology, our tools, to look if we can achieve some better results with respect to currently employed practices. And so we can both benefit from this pilot study. You can find maybe a more promising solution for your problem, and we can find a promising application of our technology. So with this, I'm concluding my talk. Thank you for your attention. Here you can find our website, genogra.com, and our email contacts. So just drop us an email if you want to collaborate or we are interested in investing in us. Thank you.
Thank you for sharing GenoGra with us. I have a question and it's related on the difficulties that you have faced to bring research outcome to the market.
Oh, this is a very interesting question and probably is one that I cannot answer in a complete way because we are not on the market yet. So we are still in the process of this technology transfer process. So we are trying to bring the results of our research on the market. And I think the most important thing is for a researcher to be an entrepreneur is a sort of change of mindset. So as a researchers, we are often involved in some activities that allow us to bring new knowledge to the world, to advance the state of the art of just a tiny bit, but it's very important. It's a very important contribution. And we always tend to, let's say, sell our work just after achieving the results. We do the work before, and then we try, for example, to valorize it with a publication and so on. What you need to do when you move to the entrepreneurial world, let's say, is probably start selling before developing. And this is a complete change of mindset that is not easy for us researchers because we are not feeling okay if we have not developed the technology that we are trying to sell. But actually, in a startup creation process, probably you need to find the product that can be sold and then starting developing it. So is this change